This is a video for the IB Biology course for topic 1.3. So as you can see here, we've got all our understandings, applications and skills for topic 1.3. They're all about membranes, membrane proteins, and the other things you need to learn about membranes. So first off, membranes in cells are composed of small building blocks like these called phospholipids. They are comprised of a hydrophilic head and a hydrophobic tail. So the hydrophilic head is made up of a phosphate group and a glycerol, and the tails are made up of fatty acids, so long chains of carbons with hydrogens branched off of them. This arrangement of hydrophobic and hydrophilic in the molecule leads to the molecules arranging in a bilayer form when exposed to water, as the hydrophilic heads are attracted and therefore go on the outside of the membrane, protecting the inside hydrophilic areas from the water. Inside membranes, you've got plenty of other things on top of phospholipids. So one such component is cholesterol, pictured here, a molecule of cholesterol, and also here in this diagram. So it slots in next to a phospholipid, and it has the same sort of structure of a polar hydrophilic head and then a hydrophobic tail that slots inside the waterless environment. So cholesterol is a key component of animal cell membranes. It's used to regulate membrane fluidity. It reduces permeability to some solutes, and allows for membrane function at a much wider range of temperatures, which is very beneficial to cells in an organism. Plant cells, on the other hand, have cell walls, and they regulate their membrane fluidity with saturated or unsaturated fatty acids. On top of cholesterol, you'll find membrane proteins inside a membrane, so these other pieces here. Membrane proteins are embedded into the membrane and they perform a specific function. There's always a reason there's a membrane protein in the membrane. They can be described as integral or peripheral, shown here. So integral or transmembrane goes across the full length and the full width of the membrane, and peripheral proteins sit on top or just embedded into one layer. So a few of the functions that you'll find membrane proteins are for transport, moving things in and out of the cell, enzymatic activity, signals such as in nerve cells, cell-to-cell recognition with glycoproteins on the outside, intercellular joining or attachment. So very wide range of functions, but all very important to sustaining the life of an organism. So the model we've been talking about for this cellular membrane is called the Singer-Nicolson model, or the fluid mosaic model, but in fact it's not always been the accepted model for membranes, with the Davison-Danieli model also being used in the past. So that was composed of protein on the outside with a phospholipid bilayer, and then protein again on the other side. So the reason they thought that might be the case is due to electron microscopy, where taking a cross-section of the membrane revealed a dark layer on top and a light layer in the middle. So in electron microscopy, proteins show up as dark areas and lipids show up as light areas. So that is how they arrived at that conclusion. Over here, though, we see the problems with the Davison-Danieli model. It assumed all membranes were equally thick, It assumed they were all symmetrical. It didn't account for the permeability of many subsystems, as they would have to go through the protein layer, and the temperatures would not have worked because it would have been solidified. The evidence that led to the Singham-Nicholson model being proposed were that membrane proteins were discovered to be insoluble in water, making them being on the outside very unlikely in a water-based environment. And also fluorescent antibody tagging of membrane proteins showed they weren't fixed in place. And freeze fracturing revealed irregular rough surfaces within the membrane. So freeze fracturing is cutting the membrane in half. And that showed that it was not uniform protein placement. The final thing is just to remember this drawing. If you're asked to draw a fluid mosaic model in an exam, it doesn't need to be too complicated. Just 2 rows of phospholipids, so the bilayer, and then a couple of proteins, and it shows some cholesterol as well. But yeah, that is everything for topic 1.3.